Hello, my name is Yasmin Panchbaya and I am a Product Technical and Customer Service Support Associate at Bio-tek. I'm going to give you a quick overview of one of our application notes detailing the performance of our E.Z.N.A soil DNA kit when extracting DNA from soil samples. This paper details how the molecular analysis of soil DNA offers a direct solution for detecting microorganisms residing in soil and studying microbial diversity. Isolation of DNA from soils is often challenging because of the presence of many contaminants that can interfere with the extraction process and are inhibitory to several downstream applications. An ideal DNA extraction method should effectively eliminate inhibitory substances and maximize DNA yields. The main objective of the study was to compare the performance of Bio-tek's E.Z.N.A soil DNA kit to that of Company M's soil DNA isolation kit in terms of DNA yield and quality. Total DNA was isolated from outdoor soil spiked with Zymo Biomics microbial community standard from Zymo Research, which is comprised of 10 microbial strains and serves as a benchmark for comparison. Extraction was performed using our E.Z.N.A soil DNA kit and Company M's DNA isolation kit. The soil samples were homogenized and the isolations were done in triplicate. Here you can see an illustration of the general protocol. The extraction protocol times were approximately 60 minutes and 70 minutes to Company M and Bio-tek respectively, and the ease of use was comparable. The DNA yields from the soil samples spiked with Zymo Biomics using the Omega kit and Company M's kit are shown in Figure 1. The performance of the Omega kit was significantly better compared to Company M's, with a 40% increase in yield when eluted in 100 microliters final volume. The quality of the DNA obtained from each extraction was determined based on the CT values generated from a qPCR reaction. Table 1 shows the average CT values obtained. The CTs seem to be slightly lower with the Bio-tek extractions indicating higher quality. Figure 2 shows average CT values obtained using bacteria and yeast-specific primers using 10x and 100x diluted purified DNA as a template. CTs were lower by 1 cycle for the gram-positive bacterium and almost 3 cycles lower for the yeast strain. That is a 2-fold and 8-fold higher yield with the Bio-tek kit when compared to the Company M kit. This data suggests that Bio-tek's kit performed as well as the Company M kit in eliminating PCR inhibitors from the isolations, but with superior yields. Overall, the results show that Bio-tek's E.Z.N.A soil DNA kit isolates high-yielding, high-quality DNA compatible with various downstream applications such as qPCR and next-generation sequencing. Thank you. We hope you find this application note to be a useful resource. Be sure to visit our website to download a copy of the application note reviewed today.